address human gut microbiota and their metabolic end products using 16S rDNA sequencing and short-chain fatty acid analysis in Indian population. So let's begin with the introduction. What is human microbiome and its importance? Basically, the bacterial communities that live in or on the human host, collectively known as human microbiome. The most common niches for microflora is the GI tract of human, and these microbes plays a really important role in nutrition, immunomodulation, and they also helps in maintaining normal intestinal homeostasis in human. So why this intestinal homeostasis need to be maintained? Because any changes in the microflora can lead to the imbalance in the metabolic products and bacterial toxin compounds, and this ultimately leads to the disease condition. But the problem that comes in the conventional culture dependent technique is that only 1% of the microflora is cultivable. So the newer approach has been used for the analysis of microbiome of an environment, and with the help of this advanced high throughput sequencing technology and advancement in the bioinformatics, now the analysis of microflora of a non-cultivable gut microbiota is not possible. And the researchers around the globe has accepted this as an efficient approach for the analysis of microflora. But we know that the composition of microbiota changes with the dietary condition and the disease condition. So the studies that have been conducted in the western part of the world cannot be extrapolated to their Indian counterparts. So there is a need for extensive study of the Indian population so that comprehensive and representative data set can be obtained. Here we can see the both the approaches, metagenomics and the conventional genomic approaches that can be used in the lab conditions. Here we can see different niches for the microflora are available, but when it comes to human gut, at the strain and species level, a huge diversity can be seen. Here we can see that human gut microbiota majorly comprised of bacteroidetes and firmicutes, and other phyla are proteobacteria, actinobacteria, fusobacteria, etc. So the hypothesis of the study, the human intestinal microbial community is quite unique and dependent on the dietary conditions and health conditions. So it does not change much except in the disease condition or in the use of antibiotics. Reports have been suggested that unique genomic or functional fingerprints of individual gut microflora exist. So this is the method that we followed. First we enrolled the patient and they collected their stool samples from 4 specific groups: vegetarian, non-vegetarian, IBD patients, and colon cancer patients. So this is the methodology. Genomic DNA isolation from fecal sample was done, and then 16S rDNA PCR amplification was done, and then we prepared the clonal libraries, and then ligation and transformation of 16S rDNA gene was done, and then we screened the clones by using amplified ribosomal DNA restriction analysis. Later we carried out the sequencing, and analysis was done using RDP, Ribosomal Database Project. This is the pictorial representation of the method that we followed. This is the gel picture of the genomic DNA isolated from fecal sample. This is the 16S rDNA PCR amplification was the gel picture showing. And this is the gel picture of the clonal library that we prepared. And a screening was done by using the ADRA technique using HEI3 as a restriction digestion enzyme. And we also did the short chain fatty acid analysis by gas chromatography. Chromatograms were recorded and peaks were analyzed. Now comes to the result. These are the patients that we screened through ADRA. 1 from vegetarian, 2 from non-vegetarian, 3 from IBD patient, and 1 from colon cancer patient. This is the RDP profiling of the gut microflora through sequencing of rDNA clonal libraries. Here we can see that IBD patients had higher percentage of bacteroids than firmicutes. And we also observed that the ratio of bacteroids and firmicutes was lower than the healthy adults. Here we are talking about the non-vegetarian healthy adults. And here the comparison shows between the colon cancer patient and healthy adults. Here we can see at the class level, Clostridia was much higher than the healthy controls. This is a pictorial representation of the same, where D2 is the colon cancer patient, and we can see that the Clostridia is much higher than the healthy adults of B2. Here we can see, here we did the blast and we got certain Clostridia sacromyticum from different ID of the patients. But the Clostridium septicum, which known to be associated with the colon cancer, was not found. The short chain fatty acid analysis profile, here we observed that there was no significant difference between the healthy controls, but when compared to the IBD and colon cancer patient, concentration of fatty acid was much higher in healthy controls. FAME analysis showed significant lower level of butyric acid in IBD patient when compared to the healthy controls. This probably could be attributed to the diet of the diseased patient, which might be rich in lignocellulosic content. This is the table showing the comparison of SCFA data composition of stool sample was done by the enrolled patient. Now comes to the summary and conclusion. Genomic profiling of all groups show remarkable difference with unique difference diversity attributed to Indian diet. The sequence cloned from adult groups belong to the proteobacteria, bacteroides, firmicutes, actinobacteria, and vericomicrobiota phylum. Recruited IBD patients were majorly from the remission phase. Clones from sample of IBD patients fell primarily into the phylum Bacteroidetes, followed by vericomicrobiota and firmicutes. The IBD patient had higher percentage of bacteroids than firmicutes, but the lower ratio of bacteroids and firmicute than the healthy non-vegetarian adults. Results indicate that microflora of a healthy adult showed high number of bacteroids and less of firmicutes. The most commonest bacterial phenomena from the sample of colon carcinoma patient was bacteroids, followed by firmicutes. There was a difference in the class Clostridia, which was 2% in healthy individuals and 24% in colon cancer patient. FAME analysis showed significant higher level of butyric acid in healthy controls, which can be indicative of lactobacilli metabolism. Propionic acid, which in conjugation with butyric acid exert anti-proliferative and anti-inflammatory effects in colon, was found lower in both IBD and colon cancer patients. Presence of increased level of isobutyric and isovaleric acid associated with disease condition due to increased colonocytes turnover along with the increased shedding. Significant lower level of hexanoic acid in both IBD and colon cancer was observed. Presence of gut microbial dysbiosis in the form of reduced proportion of bacteroidetes with respect to the firmicutes in IBD patient, even during the remission phase, highlights the role of microbial diversity in pathogenesis of mucosal inflammation in IBD. It supports the hypothesis that each individual has a unique gut microbial diversity influenced by food habits and health status. Future studies are needed to provide deeper coverage of the microbiome and to assess the effect of age, diet, and pathologic states, which can provide broader view of human biology, including new biomarkers for defining our health. Thank you.